here. And how basic science findings could be used to develop novel and innovative approaches to study antimicrobial resistant pathogens. As you know, I am an infectious disease physician, and so I'm always thinking about how to help my patients and evaluate this concept of individualized medicine in infectious diseases using translational strategies for antimicrobial resistant organisms. I forgot to mention, I do Twitter. This is my Twitter handle. If you want to follow me at superbugdoc. So this is where I was born. This is the capital of Colombia, Bogota. Is 2,600 meters above the sea level. We call it 2,600, closest to the stars. You know, I was raised, born and raised there, and I grew up in a very interesting times in the 1980s with all these movies that you talk about and series, and it's actually true, most of those. So that's what I did my medical school, and you know, and I think I learned a lot very well how to treat patients, you know, a good medical education, but you know, the limitations on the environment and the social situation. The universities, particularly medical schools, don't really have a pathway for you to follow research. So even if you had some curiosity, and that's what I had at that time, I wanted to just try to take advantage of that and say it. So at the last year of medical school, I had the opportunity to have an exchange program with the University of the United States in Boston. And that sort of opened my eyes to research and the possibility to do research. So then I jumped from there to Europe. I landed in the UK. This is the London Royal Hospital in East London, where I actually did, you know, I became interested in antimicrobial resistance since early age. I came back from the United States. I did a social service year in surgical microbiology, working with transplant surgeons. And then I became interested in microbiology and mostly bacteriology and how that influences health and all patient care. So I decided I wanted to live in Europe. And you know, I had the opportunity with a scholarship to move to England. I did a master in clinical microbiology at the University of London in this hospital, where I met these two people. So it's David Livermore, who is still known, a very known experts on gram-negative bacteria, and Lucinda Hall is a molecular microbiologist, where I started understanding basic science, basic techniques in molecular microbiology, molecular biology, and cloning, and all this, which was very interesting. Then this man, Dr. Peter Reynolds, came to give a talk in the Masters from Cambridge. This is the Department of Biochemistry, Tennis Court Road in Cambridge. This is the Hopkins building. They have another building called the Sanger building, with Fred Sanger won 2 Nobel Prizes. So this guy came to give a talk, and he talked about VRE and what are the mechanisms of resistance to vancomycin resistant in Enterococci. And I was absolutely fascinated by the incredible strategies that these organisms had to become resistant to antibiotic. And I fell in love with this topic. And this is what I wanted. I had the opportunity then to move to Cambridge to do my PhD. And I worked with this to elucidate a new metabolic pathway for vancomycin resistance. And in the interim, I had the chance to work with Professor Patrice Courvalan at the Institut Pasteur, who you know, is a very well-known expert in mechanisms of resistance. He describes several mechanisms of resistance. And one of the first people who actually understand or understood the movement of mobile elements in bacteria carrying antibiotic-resistant determinants, and the flow of these genes from gram-positive bacteria to gram-negative bacteria. So that was a very exciting time. You know, a lot of learning and new experiences, and that basically shaped my career. At the end of my PhD, I had to return to Colombia, which I did, and I founded this unit with a Wellcome Trust grant, but I was missing patient contact, and I decided then to do my clinical training And I applied in the United States because I thought that the programs of training in residency in the United States are really superior to and learn medicine and patient care, and then apply things. And I really wanted to continue to work in these organisms, vancomycin resistant enterococci, mechanisms of resistance, and try to find somebody in the United States who learned, who knew, who were funded for this. And there were actually two major players. One was in Boston, who was actually Barbara's mentor at that time at the Deaconess Hospital. And Barbara, who at that time, and this is Barbara Murray, so she became my mentor. She was very well funded and very highly recommended. And this is the reason I ended up in Houston, Texas, about 18 years ago. My God, that's a long time. So I did my clinical training in UT with MD Anderson in internal medicine and infectious diseases. And then I started, continue working on enterococci, a little bit of pathogenesis, that what Barbara was doing, but then eventually, returned to really antimicrobial and mechanistic basis of resistance, and then moving towards a more patient-oriented research, and that's what I'm going to try to present in the rest of the time that is left. So this is one of the things why I think this bug is fascinating, and why antibody resistance is fascinating. This is a Petri dish. These are antibiotic discs. As you know, antibiotics diffuse and should inhibit bacteria. In this case, you have a Petri dish, and what you have here is this halo is not an halo inhibition. It's actually bacteria growing around the disc of vancomycin. So this bug, particular bug, who was isolated from the blood of a patient, could only grow in the presence of the antibiotic, right? So this is kind of the maximum expression of resistance. Not only the bug was resistant to the antibiotic, but became addicted to the antibiotic. So it's really drug addiction in bacteria. And you actually remove the antibiotic and the bacteria kind of grow. And in the patient, interesting, the antibiotic was stopped, and bacteremia, the bloodstream infection was clear, but they mutated and they become independent of the antibiotic. So these are the sort of things that bugs can do. These are particular enterococci, so that I became fascinated with that, trying to understand how they do it, to see if we can design strategies to counteract it. This is, for example, one of the first description of Enterococci causing endocarditis. Look at the paper, it's 1899. There was no antibiotics at that time. This is a beautiful description in the Osler service at Johns Hopkins, where you read the description. This organism ended up in the heart valve, seated there, caused an infection. There was no antibiotics at that time. The patient actually died of this. Obviously, they did a pathological description of the valve. They isolated the organism, and they call it Micrococcus simogenes, but it's actually Enterococcus fecalis nowadays. They develop animal models and all this. This is 1899, no antibiotics, dead. This is actually a fellow that now is in Chile, Jose Munita. This is a patient now in MD Anderson Cancer Center around 2013. Who had a bloodstream infection with an enterococci, in this case Enterococcus fission, was treated with the top antibiotics available at that time. The patient actually, these stars represent bacteremia, so where the blood culture was positive. However, after 3 months, impossible to deal with this, the patient actually deceased. The same data of the death, the patient blood cultures were positive. Most importantly, or as important, is that during the course of this human experiment quotation, the organism became from susceptible of these two antibiotics to resistant. So these MICs from 3 to 256, 1.5 to 64. So this is the really human toll of resistance when you try to give antibiotic that doesn't work. And if anything, this pandemic taught us is that you don't have a antimicrobial, antiviral, antibacterial against an organism, and this organism is highly virulent or can cause death, there's nothing much we can do. And we experienced that during this pandemic. We've been experiencing that with superbugs for a long time now, because we are now facing patients we don't have really alternatives to treat. So we have to, come up with innovative ways to do it, to try to do something about it. So in fact, for this organism, and this is just an example of one of these organisms, there is only one drug that is approved by the FDA. It's called linezolid. This is actually a study performed with a VA cohort retrospective that actually showed that linezolid was actually not as good as this other antibiotic that is not approved for enterococci, but has very good in vitro activity, and this is called daptomycin. This is cases, so clearance of bacteremia in this case, and it's also correlated with mortality. Daptomycin is able to clear more patients than linezolid in a significant manner. And since these studies, really, daptomycin has become like the go-to drug to treat vancomycin-resistant enterococci that is resistant to other drugs. So we've been studying this drug for a long time now, almost, probably 7 years, almost 10 years. And this has given us a very interesting insights in how bacteria respond to antibiotics, in particularly how they adapt their cell envelope. And I'm going to tell you about the story in a minute, because this drug has become like a tool for us to understand bacterial evolutions in the presence of antibiotics. So daptomycin is a lipopeptide, so has different amino acids and fatty acids that usually binds calcium for its antimicrobial activity. So it becomes a de facto cationic antimicrobial peptide. So this is similar to the antimicrobial peptides, for example, that are produced by immune system. This is L37, that usually target the cell membrane of invading bacteria. And this is important because some of these mechanisms are related with the evolution of the interaction between the immune system and the pathogen. Daptomycin, the mechanism of action is actually not well understood right now, but we have more insights right now that has actually rebranded our view of how this antibiotic kills bacteria. This is the model of daptomycin binding to the cell membrane. These are phospholipids, and one of the phospholipids that is necessary for its action is phosphatidylglycerol. So this drug actually targets the outer membrane of the cell membrane, initially in bacteria, and appears to oligomerize and going to the inner membrane, eventually causing major changes in cell envelope homeostasis. So recent studies by a German group, he actually gave a talk in our symposium last week, suggested that the first daptomycin targets fluid domains in the cell membrane, and when it oligomerizes from the outer membrane to the inner membrane, basically displaces important protein in cell wall metabolism. This is, for example, MURG, that is important for the synthesis of lipid-2 antibiotics, but also other peripheral proteins that are involved in phospholipid metabolism. And more recently, this group showed that daptomycin, depicted here in red with calcium, actually is able to bind peptidoglycan precursors inside the membrane by binding to the bactoprenyl couplet lipid intermediate from the sort of a tripartite complex between lipid-2, C55, and the antibiotics. And this is sort of the PVPs and potentially other mechanisms, displacing also more MURG. So this all suggests that this drug, even though it acts at the membrane level, disrupts cell wall synthesis, also disrupts cell membrane homeostasis. So it's a massive hit to the bacterial cells, and that's why the mechanism of action is there. And it's very similar in a way, in the approach that antimicrobial peptides produced by the immune system actually kill bacteria, because they also interact in the membrane, and most of them, or several of them, interact with lipid intermediates that are necessary for peptidoglycan synthesis. And without cell wall, bacteria are not very fit and are likely to die under the influence of the immune system. So, the mechanism of resistance becomes really interesting, because bacteria will eventually have to remodel the cell envelope for this, but there may be adapted to do this. And this story starts more or less about 10 years ago or so, with a patient that was treated with daptomycin and had an organism that was susceptible and then became resistant during therapy. And we know by whole genome sequencing that they were identical, was the same strain, but we actually found only 3 deletions, three changes in genes in this, and these are depicted in this slide. 2 of these are related with phospholipid enzymes, which I'm not going to comment anymore in this talks for the sake of time. And one is this called LIA-F. This is to introduce you to the LIA-FSR system, which is one of these systems that bacteria use, particularly gram-positive firmicutes, to protect their cell envelopes. It's a 3-component regulatory system, usually a membrane protein, as histidine kinase bound to the membrane, and then a response regulator. And in absence of antibiotic, there seems to be an interaction that has the system off. But under antibiotic attack, in this case, for example, daptomycin, there is probably an interaction, the LIA-F and LIA-S, either inhibiting or activating the system. LIA-S as a sensor of a damage of the changes produced by the antimicrobial. Phosphorylated response regulator activates a bunch of genes. And you can see how this happens. This is actually pictures from the same strain before and after the antibiotic, S and R. And this is staining with a stain called nonanyl ochrine orange, which actually causes, in this case, green fluorescence when there are anionic phospholipids. And these anionic phospholipids, for example, phosphatidylglycerol, cardiolipin, tend to concentrate in the septum and are actually important for cell division and some in the poles. When you see in the resistant strain that you see the pattern of this staining completely changes. Cells don't divide very well. Most importantly, you don't see septum there. And using a simple allelic exchange, we could make a susceptible strain, putting an allelic strain of this protein LIA-F that encodes this transmembrane protein upstream of the LIA-FSR system, and actually make the same pattern of changes in the cell membrane of the bacterium. So, clearly that system is in charge or associated with the regulation and control of the cell membrane envelope. So the first target that we had to characterize was the response regulator. And this work has been done with my main collaborator here is Dr. Yusuf Shamu at Rice University. So this is the crystal structure that his group obtains in collaboration with us of fecalis and fission LIA-R. This is a receiver domain and DNA binding domain. This is the aspartase with is phosphorylated. But we found in the several clinical strains that, for example, these aspartates are mutated, or there are other mutations that actually make this response regulator very active even in the absence of phosphorylations. And in fission in particular, there are several epistatic interactions, mutations in LIA-S and LIA-R that actually favor this state. So this response regulator seems to be a very fast and very effective way for the cell to respond to the attack of antimicrobial peptides and antibiotics. So this is the DNA domain that basically, when it's activated, oligomerizes, and we know in fecalis is a tetramer and fission is probably a dimer, interacting with a specific basis of the DNA. And Dr. Chamu in his group mapped this, promoted very well, and with an extended binding site suggesting, important oligomerization of this, with nucleotides that really interact with with this response regulator, and allow the cell to respond to the antibiotic attack with certain genes. This is a model when the RNA polymerases then can activate quickly and bend the DNA in the presence of a tetramer of the response regulator. This is one of the mutations actually responsible for this. It was found in evolution experiments that actually does not require phosphorylation, so allowing bacteria to respond quickly. So in the last few years, we have been dedicating to understand then what are the target genes of this response regulator. This is Aisha Khan who actually performed this, and Cecilia Tran this. So so we found that one of the main targets was a cluster of 3 genes. I'm going to tell you about this gene encoding a protein of 533 amino acid called LIA-X, with two domains, alpha helix N-terminal and a beta sheeted pleat C-terminal domain. And one of the interesting things that happened is that this is the same now staining, and now we're seeing simple cells. This is the wild type E. faecalis laboratory strain. You see the now stain in the septum as it should be. But when we produce sort of deletions of the C-terminal of this protein with a truncation, then we see this rearrangement of these phospholipid microdomains, suggesting membrane adaptation, and actually we obtain an increase in MIC to the resistant levels. That with the C-terminal, or even with the entire gene that coding this protein, we see the same effect. And you can see here how we measure, mostly in the septum. This is in different parts. There is not a real pattern. And when we complement, we can complement this and back to the septum, as you can see the fluorescence. So clearly this protein was at the hump and controlling at the membrane level the distribution of cell membrane microdomains and remodeling. So Aisha tried to identify where this protein was, and each color represents a set of strains. This is the wild type OG1RF, and you see that when the cell becomes resistant, she can find it a lot in the surface and in the extracellular surface. And these are blue and green represent clinical strains compared to the susceptible isolates. And she can also find a lot of this protein both in the surface of the cell and extracellularly. And she did some gold staining of the protein, and she could, in the resistant derivative, she can find a lot of this protein in the extracellular domain. So this protein, when the situation is activated, will be exposed on the surface and then released to extracellular medium. So what was the mechanism? That's the one that always have driven me to understand this. The first thought that is that this protein actually binds the antimicrobial peptides or the antibiotics and prevent them to reach the membrane. And we have some evidence for that. This is microscope photophoresis that, again, with Dr. Shamoud was developed. Aisha did several experiments in which that she showed clearly that here the change of fluorescence of the interaction tells you that there is interaction. So you can see interaction here either from the full protein or the N-terminal as you increase the concentration until there is a plateau, suggesting there is interaction and binding of LIA-X with daptomycin. And not only that, she also did this with LL37. And as you increase the concentration, you can see the fluorescent changes, and there is interaction in this curve, suggesting also interaction of this protein called LIA-X with extracellular peptides. So we thought, okay, so this looks like an anti-missile. So if the antibiotic is a missile, this should be an anti-missile, and then it should prevent the arrival of the antibiotic to the target, which is the cell membrane. So in order to prove that, Aisha took a supernatant of resistant strains, for example, and tried to do MICs with this supernatant for susceptible. If the mechanism was as such that you would prevent the antibiotic to reach the membrane, you should be able to see increases in the MIC with the supernatant of resistant strain. And that's in fact what she saw. So she saw, for example, if you do an MIC on a susceptible strain with a resistant supernatant, you can see increase in the MIC. These are the same things there. But interestingly, when she did it in the absence of the response regulator, there was no response. She also used the purified protein and saw a similar effect. You add the protein, you increase the MIC to levels, but that increase doesn't happen with the knockout of the response regulator. nor happen in other species like in Staph aureus. No, so in Staph aureus, we could not see that effect. Which actually suggested that this is actually a very specific system in Enterococci that is dependent on LIAR. And although it binds the daptomycin and antimicrobial peptides, this conjugate between these two molecules need to get back to the membrane and trigger a signal cascade. So this protein not only controls at the membrane the redistribution of phospholipid microdomains, but serves as a signaling molecule to basically patrol the extracellular milieu with that is full of antimicrobials or antimicrobial peptides. So this is a novel mechanisms, and in order to close this story, Aisha used with the collaboration of Danielle Garcin in a C. elegans, she showed that strains that actually had the mutations or were resistant were more virulent, either live strains or clinical strain. But that virulence went away when you use hosts like these worms called PMK1 worms. And so this virulence advantage was completely abolished in these cases. So that suggested that this is a specific mechanism by which what this superbug, sort of screens the environment and can detect antimicrobials and antibiotics. So this is the stay off. LIAX with these two domains is in the surface at low quantities. We think when the system is on, oligomerizes, and then the N terminal sort of is cleaved and can detect LL37 and daptomycin or other antimicrobial peptides, and then maintain the system on in order to adapt the membrane, and this C-terminal controls membrane phospholipid rearrangement that eventually is going to alter peptidoglycan synthesis as well. So very clever and complex way to defend, but now we have ways to potentially do that and target this system, and we are developing innovative approaches to try to reverse this resistant phenotype. So cool findings, but how do you apply this to patient care? That's at the end of the day, this is what I care. So the first thing that you need to know is that every single bacteria is put through a system in the lab, in the clinical microbiology lab, that is going to tell you which antibiotic is susceptible or resistant to, and then you, as a clinician, you choose these antibiotics to treat the patient. That's called minimal inhibitory concentration, and that's a routine laboratory method. However, for this particular bug, is very unreliable for many reasons. This is actually a study that we did with the 3 most famous labs in the country, led by Dr. Romney Humphreys. She sent these organisms to different labs. Each bar is a different lab, and these are the values that came back. As you see, there is a lot of discrepancy here. So this MIC is not reliable. So how can you treat a patient if your main sort of source of information is not reliable? So that's what we tried to do. So there was initial observation that suggested that this protein that I just told you about, for example, YIX, we can detect and develop an ELISA. And as this is actually different strain from a patient that also was susceptible and then became resistant, we could detect increased quantities of LIAX. So we thought this could be used potentially as a surrogate marker to even drop MICs that are so unreliable. So Deidre Axe-House, who is our T32 fellow, you saw her in the meeting last week, and Cecilia Tron, they've been testing this hypothesis using a collection of clinical strains. This is initial preliminary results that seems to correlate with the MIC. Most interestingly, you can see MIC that potentially are depicted as susceptible, but have high MICs, high detection of LIAX, suggesting that they might be tolerant to the antibiotics. So we are currently, and this is one of the Deidre's project, to try to develop a MIC independent method to just test susceptibility, not only in bugs, but you could potentially use it in clinical samples, even in stool samples, etc. So that will be one of the translational applications of the findings here. Um, we if we want to do that, we also want to have clinical data from patients. So this is a project that is about 2 years now is led by a fellow in infectious diseases at UT, Herman Contreras, and also Jose Munita and Cecilia Tran. And this is called a Venus project. So this is a pure clinical prospective study that collects strains from different sites. We have sites in the United States, Argentina, Chile, Spain, Germany, Brazil, and South Korea. We actually collect the isolate, collect the clinical data. All the isolates get subject to whole genome sequencing. But contrary of any other study, every patient that is included in these database needs to have a follow-up blood culture, which in infectious diseases is very important. You have a bloodstream infection, you want to show potentially that your blood is clear, particularly in gram-positive bloodstream infections. So that allows us to, for the first time, evaluate microbiological outcomes in these patients. And this is just a summary of the first patients that it is. But what we found is the lack of eradication of the bloodstream from a patient like this is highly associated. This is a multivariate analysis with mortality. So you don't clear the organism from the blood, the patient has high odds of dying. And apart from neutropenia, which is another very important factor, it was very highly associated, related with mortality. So in order to clear the bug, you need to have the right antibiotic, and you have to have the right test to do it. And we don't at the moment, and we think we can do much better than this. He could show, for example, that mortality changes based on the phenotype of resistance. Mortality is not the same. It's very early here. This is Kaplan-Meier curves that show survival curves, and it's clearly that the presence of these organisms is probably associated with a bad prognosis in patients. So if you want to clear that, how do you do it? So daptomycin, and our clinical experience is you use daptomycin, you still have patients that fail. So this is actually another translational aspect with pharmacologists. We actually pair with Dr. Mike Ryback, who is a PharmD who specializes in a model called subendocardial vegetation model. And you can test humanized doses of the antibiotic. So these are, for example, different doses of daptomycin, and this is a model that you can count colonies through time. As you can see here, in these doses, for example, there is no bacterial killing. In each arrow represents the isolation of a resistant derivative. When you increase the MIC to 10, sorry, the dose to 10 milligrams per kilo, then you see some killing, but you still have resistance. So this is the best antibiotic we have, and this is most likely what happens in patients. So it's really suboptimal. But we found that for several observations that you add a beta-lactam, for example, ampicillin, even at low concentration, you got very good killing, and then you lack resistance. And this is something that needs to be tested, that we want to test in humans, in clinical trials, but also understand more of the mechanistic basis of this interaction called a Cesar effect, that the combination is very potent between daptomycin and beta-lactams. Even for resistant strains, this is a resistant strain with daptomycin alone. This is with the same model with ampicillin at different concentration. And we could show in an animal model, for example, as you can see here, I believe is the blue one, the combination of daptomycin and ampicillin here. You can see these are a number of bacteria in the vegetation, the heart valves. This is the controls with different antibiotics. And this is, as you can see, the largest decrease in bacterial burden in this vegetation with the combination of DAP ampicillin, and DAP ceftadolin. Those are the only ones that actually had what we call a sterile vegetation at the limit of detection. So we can, from all that, get to interesting questions, and from the patient back to the lab, and the lab back to the patient. So I also wanted to tell you a little bit as the objective of this, of some of the international work with this. This is the international unit that we have in Colombia, Bogota, Colombia. Several members of my lab had visited this. This is a hospital that was open about 2 years ago, where it's open also for collaborations there. This is a case of the first case of a patient in Colombia that had an infection called methicillin-resistant community-associated MRSA in Colombia. It was a dancer, capoeira dancer, that actually had this horrendous infection. This is the chest during surgery. They had multiple grafts, and he could walk but not dance again. And this was our discovery, and that time pulse field electrophoresis that showed that this strain in Colombia was very related to a strain that was disseminated in the United States, a Staph aureus called USA 300. They were related, but they were different sequence type, and there was a difference in some of the subtypes that carry antibiotic-resistant genes. Most importantly, we started following this in Latin America, and this is Ginette Reyes that now coordinate the unit in Colombia. And you see this strain was circulating in 2008 in a high proportion, both in Ecuador and Colombia, shown here in red. And then part of this is launching the genomic mission that we have tried to in the last few years. This is with Paul Planet and George Wyston, who used to be the leader of the microbiome project at Baylor College of Medicine, and basically show that this is North American and South American strains are very related, and are probably with an early branching common ancestor. And using a molecular clock, we could determine that this strain, more or less in mid-70s, sort of branched out, one acquiring an island that is called ACME for arginine catabol metabolic elements in North America, and another one called Comer in South America. And this Comer island is called that for copper and mercury resistant genes. And we know that the South American strains have an important component of that. And of note, Bogota's water has one of the most highest concentration of mercury in the world, and there's contamination for mining and other activities there, putting a context, environmental contamination with antibiotic resistance and dissemination of that, and we are actively studying this thing. In the last few minutes, I want to talk about a project that it comes from a very simple observation, which is called the Inoculum Effect. And this is the use of cephalosporins in the treatment of staphylococcal endocarditis. And the observation that in certain organisms, this cephalosporin fails and is due what we call the inoculum effect. And inoculum refers to high inoculum bacterial inoculum, but this is likely related to one factor, which is the presence of beta-lactamase. This is Will Miller. This is one of his studies. He helped us with this study. This is in Argentina, where they only have cephalosporin, and we found that it was associated with mortality. This is Dr. Maris, Barbara Maris, my mentor, work in which she could clearly, and Kavindra Singh, they could basically show that you cure the beta-lactamases from the staph, then you don't produce an effect. This is an endocarditis model. This is the number of colonies, and as you can see, if you have the beta-lactamase, the counts are much higher than if you cure it. So this is due to the beta-lactamase. So this beta-lactamase is produced in the cytoplasm, is bound to the membrane in Staph aureus, and has two forms: one that is released to the extracellular space, and then one that remains on the surface as a lipoprotein. So we made a very simple observation that the release of the extracellular beta-lactamase was actually associated with failures and with the inoculum effect. And just with that, we just have developed a test that we think can be in 2 hours, genetic with part of Latin American strains. Now we're testing this in the laboratory We know this test is about 80–89% in sensitivity and specificity respectively, but with the most common beta-lactamase, it goes up to the 90s. And now we're testing in the field to try to improve the treatment of Staphylococcus aureus serious infections, methicillin susceptible, even though it is very susceptible. Finally, using genomics, one of our mission has been to try to understand the dissemination of this. This is one of the nanopore platforms. Again, Will Miller, this is using a patient, and this is coming directly from the patient, a patient that gets to the hospital. This is the hospital day, and eventually gets an organism that is very resistant. How do you treat that? These are the susceptibility tests. I'm not gonna stop here. What is red is very resistant. What is black is okay, but there are bad drugs that cause a lot of toxicities. Using almost real-time whole genome sequencing, we found here that this strain carry this enzyme that had not been reported here very frequently called GES, and they were in tandem, two of those. We could understand that this actually correlated a cluster with some of them circulating in Mexico. But most importantly, we could use a test in vitro that the combination of this antibiotic called ceftazidime avibactam with aztreonam produced certain inhibition. This is an E-test where the antibiotic actually diffuses. You don't see any halo of inhibition with aztreonam, but if you combine, then you see a clearance. We used it for the patient, and the patient did well. So this is really translational disease. This is, for example, another example. This is Blake Hanson, which is the Associate Director of Microbial Genomics in our group. This is with the Antimicrobial Resistant Leadership Group. This is one of the examples that of multicentric studies that we've been able to participate. I'm not gonna stop here in detail. This is a phylogenetic tree of more than 1,000 patients of Klebsiella pneumonia. But what I want to show you is this red is a genetic lineage called 258, but this actually orange, we found out that it was a particular lineage that was present mostly in Houston, and we know now Houston has a parallel epidemic of carbapenem-resistant Klebsiella pneumonia of this clonal group that we are actively studying and understanding the dissemination. Finally, with my collaborators, Dr. Sam Shelburne, and this is Will Shropshire, who is now a postdoc at MD Anderson. He did his PhD with us. Using also whole genome sequencing, he was able to understand that in Anderson, for example, several of these bacteremias are caused by carbapenem-resistant organisms that do not produce carbapenemases. These are sets of several patients, and he could using whole genome sequencing and complete assemblies, he could pinpoint that there were amplifications of this region of DNA in these isolates that actually had as low spectrum beta-lactamases like CTM15 and OXA1, and there was amplification of this of these units, randomly high amplification, 10x, 13x, 30x. But apart from that, they actually reinsert in other locations of the chromosome. In this case, producing problems in the porins and disrupting these porins, and these mobilizable units that are amplified disrupt the porins, decrease the entry of the antibiotic, and the antibiotic that comes is in lower concentration, and then all this immense amount of beta-lactamases can actually deal with that and eventually produce the phenotype. This cannot be detected in any lab. Only with this approach that we are trying to devise will be able to detect. So I have given you sort of a potpourri of how we are doing translational research, trying to go all the way from basic sciences to translational research using basic microbiology, molecular microbiology, biochemistry, structural biology, and genomics to eventually help patients and understand how we can treat better our patients. This is as the availability of these tools become apparent, we believe this could revolutionize infectious diseases, and personalized medicine in infectious diseases will be a reality in a timely manner. And I think the future of microbiology is in genomics. I believe those will be, the microbiology of the future will be genomic genomic microbiologists. So there are several people that I want to thank in my group. I also mentioned that, Diana is very important, sort of the person who actually makes everything happen. I mentioned to you several of the members of the lab. Sam and Yusuf Shamu are the main collaborators here. All the network of collaborators in the United States and globally in different countries. And of course, the funding on the support of the Antimicrobial Resistant Leadership Group, which is an NIH funded group that do all this. So I'm going to stop there and give some time for some questions. And thank you very much for your attention.
Thank you so much. This was a fascinating talk. Really enjoyed it. I'm going to start with a question from the chat. Does the LIA-R system respond to any endogenous compound in the same fashion as the antibiotics?
So endogenous compounds, if you mean antimicrobial peptides, we have tested some of them, like LL37, HNMBP from humans, some compounds from bacteria, bacteriocins, and they seem to respond the same way. So the LIA-FSR seems to be like a general respondent of the cell membrane envelope.
Okay, thank you. Is there any question from the audience? Please just unmute yourself and you can ask it or also type it here in the chat.
I have a question.
Sure.
So you mentioned that this mechanism for the daptomycin resistance is pretty unique to the Enterococcus faecalis, and I'm wondering if you know sort of why that is, or if there are analogous mechanisms in other bacteria that are not quite the same, or alternatively, like at what level of uniqueness, like is that unique to the strain level or to a species or to a genus?
Yeah.
Why is that so unique?
So it seems to be unique to the species level. So for example, faecalis, the Strygus faecalis is very similar. Fission has a different strategy. I didn't talk much about it, but instead of massive phospholipid redistribution, there seems to be changes in the surface charge that repels the antibiotic from the surface. And that's also seen, for example, in Staph aureus. We've done work in Streptococci, and we know that in Streptococci, this repulsion occurs, but there are certain cells during the population that actually seem to concentrate the antibiotic, like sort of protecting the rest of the population. So these all seem to be pretty unique. We suggest that this has been going, the evolution of this has been going for years and years. And these antibiotics are natural antibiotics, probably been around for a long time. And as bacteria become co-evolved with humans and they were exposed to these antimicrobial peptides, they develop all these different cool strategies that we are seeing. So the dissection of those will hopefully open new avenues to try to get this, to try to revert that process. Eva?
Yeah, I don't see any other questions in the chat. Is there any other question from the audience?
I guess I have a question. When you showed the graphs with the minimum inhibitory concentration, there seems to be like a very different affinity, I guess, of using the LIA protein by itself versus the N-terminus. The N-terminus having less affinity. How did you conclude that I guess that's what I got out of your model. You seem to believe that the N-terminal by itself can elicit the response, but do we see that physiologically?
Correct. So yeah. So I didn't have time to go into this in detail, but it is clear that the N-terminal is the region that is crucial for binding to sort of molecules that are attacking the cell, right? that it's likely that the protein anchors in the outer cell. We still don't know exactly how or where. That doesn't seem to be directly with the peptidoglycan, but maybe embedded in the cell wall, and maybe bound to a PVP or something, and likely is cleaved by proteases that, for example, Enterococci. We're testing that hypothesis. So the oligomerization of this protein in the attack situation allows for that to happen, and then release several of these compounds in the milieu, because you add this, just the N terminal, and that's sufficient to reproduce the phenotype. So definitely the N terminal seems to be sufficient.
Okay, we have another question in the chat from Emilio Lucero, and he's asking, is there any interaction of the DAP with the membrane of mammal cells, as these membranes also have similar phospholipids where DAP binds? Correct.
So the binding of this drug seems to be very related to phosphatidylglycerol and the concentration of phosphatidylglycerol. And that makes that certain other human cells, on even the producers or even gram negatives, so these antibiotic is not able to bind to the membrane and cause any of these effects, because the sort of concentration are much lower. However, the daptomycin, for example, can bind to the lipids or the phospholipids or the surfactant that is produced in the lungs. And that's actually a serendipity finding. They actually tested the efficacy of daptomycin in lung infection, and they realized that the antibiotic was being inactivated by the surfactant. So it was a catastrophic failure of a clinical trial, because that property was not known at that time. One of the main toxicities is muscular toxicity. That's probably related also to the lysis of some cells, but it's not clear that it's directly due to binding or something else.
Thank you. I actually have a question too. I'm curious to see how long does it take from the patient, whenever you get a sample, to get it sequenced on the nanopore, to do the analysis, to figure out what treatment you need to use? Can you comment on that?
Yeah, so normally, still, probably in different hospitals. So it takes about, since you have, let's say, a bloodstream infection, and the growth, it takes about 24 to 48 hours to know the organism, and probably an additional time to get susceptibilities. You see, if the organism grows, and we don't need a lot, I think one of these examples, we really dedicated the lab to this, and we could get samples in less than 24 hours. I think it was about 6 hours working very hard to do this. But there are technologies now that allows whole genome sequencing likely directly from clinical samples. There are even companies dedicated to that. And we've developed the pipelines, and we actually have a group, a project with the RLG called GenoSmart, to imagine to have some sort of Google thing for genome sequences, that you can input that and have an interpretation and try to give the clinician information of what genes are there or not to direct therapy. So I think that's where the field is going in microbiology, and that's what people, I will advise any of you that are interested in this, that's sort of the future for sure.
Thank you, very interesting. Are there any more questions? If not, you can also email the speaker.
I think Karen wants to say something about the innovation project, no? Dr. Karen Ethan?
Yeah, thank you. Thank you, Cesar. Thank you for sharing your journey and letting us see how you got from Bogota to Houston. That was very, very interesting. It was circuitous, but we're glad you made it. We're glad you're here in Houston. And thank you for your leadership and being the program director of our T32 grant in antimicrobial resistance. So I'm delighted that there are so many people interested in this topic of antimicrobial resistance, because it's, it's deeply, deeply important and becoming more so, by the minute practically. So Cesar was our kickoff speaker for our new series, which is going to occur during the Keck seminars for this semester and in the next, in the innovation and diversity in biomedical science. And so our goal is to be able to introduce members of underrepresented groups to share their research and their journey to show how they got into these outstanding careers in research and research related fields. Cesar, do you want to add anything to that? Anything you want to add about your journey that you can share with us besides Bogota and London and Houston?
Well, I could talk the entire hour just all the issues that happens. But one of the things that I would say is that the journey is almost the destination. You maintain your focus, and there will be moments in which everything will look very bad. But if you maintain your perseverance, and I think you always get there if you keep believing in yourself. And for a foreign medical graduate like me, to try to navigate between trying to do science, patient, and then visa, and find a job, and trying to think about just that I'm going to be kicked out of the country soon, and how I'm going to get this grant because the NIH that doesn't give me, that's daunting. I can tell you that the main issue that was critical in my career at early stages was when the NIH launched this K99R00 award, which was open for foreigners. And I was lucky enough to get one of the first K99s, which changed completely the way that I could now be settled as a faculty and then get into my career. And even though that it was not the topic I was going to research, because it was more leaf pathogenesis. So, my mentor sort of, gave me that part, but, I went back to what I wanted for my first R01. Once I used that as a platform to learn things and to understand how to do it. And I think that's the mentorship, having a mentor that can help you navigate to that is very important. And I always tell the trainees, when you have a mentor, the sniff test of the mentor, go and look at the latest publication and see how many of those he is the corresponding author. If he's always the corresponding author, that's a red flag, So that's very important, to have somebody generous who allows you to have prominence on your work and own your work. That's crucial for your development. I think that's what I wanted to share.
Cesar, what other kinds of challenges did you face? I mean, not only are you in an underrepresented group, but you're also, an immigrant. So you had to learn English. And so how did you overcome those obstacles that anyone would face moving to another country? And how did you develop your leadership skills?
So thank you, Karen. So an example of to answer the first question was I knew that I wanted to learn medicine, cutting edge. And, I was in a school that all the books were in Spanish, but I decided to have all the books in English. So I sort of learned myself to read English. I didn't speak English at all, but I knew. So at the end of my career, I was selected to have this exchange program, which is my first sort of out of the country. And they put me in a very interesting place, which is the Mass General Hospital, which is one of the meccas of medicine. And I really barely spoke English. And I know I could have done much better if I had spoken better, But with that, I think people, understood and the view that you can do that. And that I received was for the first time that I saw how science connects to patient care, because I was always thought to be a doctor, you know, to be a good clinician, but never to just think about that, to go to the lab. That was the first time. And that one of my attendings there at Maginot said, why don't you do a PhD? You know, I was really young at that time, so I had some time, and, you know, and the other two important things is I didn't have any funding, you know, and my roots are very, you know, humble. So my family didn't have any money. But fortunately, I was able to find a scholarship that took me to different places, and just taking the ability to go to different places. I mean, landing in the United States was okay because I knew, but landing in England, in London at 11 p.m. and not speaking very well English, going to a restaurant where they don't understand what you're saying, that was a jungle. That was tough, right? And feeling away from everything and all that. But I always wanted to live in Europe and all this. So in these moments of loneliness and all that, you need to keep focus on what is the main goal. There were no cell phones at that time, by the way. Email was not available. So talking to your family, you have to just stop eating. You wanted to talk to your mother. So I used to talk to my mother every month, you know, and a particular time when I knew my stomach was full. That at that time was pounds, you know, very, very strong. I mean, Colombian pesos were nothing. So yeah, so those are things that you will have to go and do it. And in that process, when you start building things, is when you start developing attitudes as a leader. Because I have been fortunate enough, I guess, to build things from scratch when there was nothing there. And you can see if you have the right people and the right attitude, you can build things. You can put things together. You can make people working together. Believe it or not, this medical center was built and still is a competition, right? And I think since the creation of the GCC, people understood that it's better to collaborate than to compete, and we all get together. But still, there is a lot of reluctance between these institutions and competition. So when you show people that you together are much better than isolated, and that everyone gets a benefit, people change their attitude. And they have to trust you that you are honest in what you're doing, and you are not taking advantage of that for your own good. And that's the way that you pull people forward, and we are together. And this is the example of this cluster. And you know, if you had the opportunity to participate last week in the meeting, I was so proud that when in this virtual meeting, we have almost 300 people plus every day in this meeting. So so this is sort of a summary of many stories that hopefully one day with a drink, I will be able to tell you more.
Well, I'd like to see if any of the trainees have any questions for Cesar about his journey. You know, feel free to speak up, and Cesar is very open and willing to share his thoughts and ideas and suggestions. So please feel free to ask him some questions.
Hello.
Yes.
Yes. My name is Ivan Coronado. I'm also Colombian as well.
Oh, well, great.
Yeah, I worked for some time in the floor under your labs.
Oh, yeah.
My question is, how difficult is to get the collaboration going between the United States and Colombia?
Yeah, that's a great question. So I was fortunate enough when I actually came as a resident here. You know, I had already have set up the basis for the lab in Colombia. So we kept doing work even though I was on call and all that. But when I was a faculty, I decided to try to use the ability to train people here and bring everybody, and actually train about 6 PhDs from Colombia here. They are all back there. So as they started to come back, and they were actually all funded by the Colombian government, called Ciencias. So they all came back and they all grew, and you know, and then the connection was very simple. So we were mature enough to have, for example, now put the institutions able to receive NIH funding, launching subclinical trials, and then expand this in Latin America. Now our unit is really Latin America. But the most important part was to get the people trained in the right way. So UT is a very Texas-centric university. You know, they have that in their mission. So you have to have innovative ways to do it. And we believe that people, for example, I have a program where I go with School of Public Health students to Colombia for 2 weeks. We go to Bogota, we go to rural areas, and they can, you know, have a wonderful experience. The clinical experience is a bit more difficult because there are certain regulations for the students and fellows. And Colombia still has a reputation in the Department of State that is not that great, so that creates some barriers. But I think we've been bringing that down. So, and I think, you know, with you can convince the institution, and there is a partner institution in Colombia that want to do things. Really, that's how, you know, elite institution has launched really good programs around the world, you know, and have units. I think this unit that is going to be 20 years last year, now we are mature enough to really be ambitious and have programs, have visitors and train fellows, etc.
Thank you.
Any other questions? Anything else you'd like to add, Cesar, that we didn't touch on? I mean, I think you were the first in your family to go to college. Is that right?
Yes, in medical school for sure. Yeah. So my family is the little town in Colombia called Girardot. They moved to Bogota. They were 10 brothers. They all worked very hard, including my mother. So the youngest brother could go to college, was the only one who go to college, was my uncle. So my father left very young when I was very young. So I don't know anything about him. So my mother's brothers and sisters sort of raised me.
Well, I'm certain they're so very proud of you.
Thank you very much. And guys, the only thing I can say is that follow your dreams. The United States is still the place for dreams, believe me. You know, people who come here from other countries, they realize there's still the countries opportunities, and continue to be plenty of opportunities regardless of the background. I think we are all very sensitive now of what it means to be an American. And I think there is opportunities like this that to share experiences, and people realize. And for me, coming from a developing country in a difficult social situation, the most important frame in my mind was to think that I was as good as anybody else, that my brain was not different from others, that I could do things the same as anybody. And was not what I was taught, or I was, you know, hard to believe. So that's the frame of mind that allows you to move forward. Okay, guys.
Thank you, Cesar.
Thank you.
Thank you very much, everybody.
Thank you.
Feel free to call or email. Okay, take care.
Thank you, everyone. Have a great Friday evening. Stay warm, and we'll see you next week.
Thank you very much.
Thanks,
Bye.
Cesar.